Welcome, everyone. Welcome to Straight Science. Straight Science is an evening seminar series put on by the University of Alaska Fairbanks Northwest Campus here in Nome and University of Alaska Fairbanks, Alaska Sea Grant. So both Northwest Campus and Alaska Sea Grant, we are public servants of the Bering Strait region. We serve all peoples of the Bering Strait region, which is the homeland and waters of the Inupiaq, St. Lawrence Island Yupik, and Central Yupik peoples. So tonight I'm very excited, and I hope they've got good news for us all. We have our two speakers are going to talk to us with an update, not just on Alaska's wildlife regarding avian influenza, but also the Bering Strait region's situation with avian influenza. Just an update on where we are and what maybe lies ahead and what we can do about it a little bit. So our speakers, and who's leading off? Andy, you're leading? Okay. Our first speaker is Andy Ramey. He is a research biologist with the U.S. Geological Survey. You heard that right. And that is the research arm for birds, and especially in your case, you are the state's authority on avian influenza in wildlife. And our next, then following up, will be Lisa Kercher. Now, this is going to be an interesting one because Lisa Kercher is a research biologist with the St. Jude's Children's Hospital, Research Hospital, St. Jude Children's Research Hospital. I've learned to say it correctly finally. And our big question is, what is St. Jude Hospital doing in Nome? So we'll hear from Lisa, and she is going to be actually right after the show going into the science lab and testing some of our birds from the region. So with that, we're really glad you're both here, and we're glad everyone's online and in the room. Andy Ramey, why don't you start us off? We're so glad you're here. Take it away.
Awesome. Thank you, Gabe. Appreciate that nice introduction. Good evening, Bering Strait residents. I'm shocked at this great turnout with this beautiful weather, so thank you all for being here tonight. I'm also thankful for another opportunity to speak as part of the Straight Science lecture series. Thanks, Gabe, for another invitation. So this evening, I'm going to provide, once again, a broad overview of highly pathogenic avian influenza, which you might know as bird flu. For those that regularly attend this lecture series, the background portion of this talk is probably going to be a review, but I have updated this talk with some additional information and included results specifically to the Nome area. Additionally, as Gabe previewed, Lisa Kercher is here tonight, and she's going to be providing details about an ongoing sampling effort here in Nome, and that should not be missed. Here's an obligatory disclaimer slide. So here's an outline for tonight's presentation. So I'm going to start with a very brief introduction of myself, my office, and the lab that I lead. Next, I'm going to provide some general background information about highly pathogenic avian influenza, or I'm going to use a shorthand for this talk and just call it HPAI. And I want to convey some of the impacts of this disease to wildlife, both in the United States and also globally. I'm then going to summarize detections of HPAI in the United States and Alaska, and again here in the Nome area. I'm going to then turn things over to Lisa so she can introduce herself, talk about St. Jude Children's Research Hospital and the sampling effort initiated here in Nome. And then finally, I'm going to conclude by providing some sources for additional information, as well as some contacts in case you witness wildlife that is sick or has died and that you think may be affected by HPAI. So I'm going to start now by introducing myself. So I was born and raised in the Midwestern US, so I've now spent most of my life here in Alaska, and I've dedicated my entire academic and professional life towards better understanding the health and status of our fish and wildlife resources. So this includes degrees in fish and wildlife biology and also veterinary and biomedical science. So I began my professional career with U.S. Fish and Wildlife Service in Western Alaska at Togac National Wildlife Refuge out of Dillingham. If any of you have been there, spent time there. And since then, I've spent the last 23 plus years living and working on traditional denied lands in Anchorage. More specifically, I spent these past 2+ decades conducting research at the USGS, at the Alaska Science Center, as Gabe recalled, and much of my research at the Alaska Science Center has been focused on avian influenza in wildlife, and this is primarily using genetic and serologic methods. Outside of work, I really enjoy hiking, skiing, paddleboarding, wildlife photography, bird hunting, and fly fishing, pretty much anything that a person can do outside here in Alaska. So as previously mentioned now a couple of times, I work at the USGS Alaska Science Center. And for those that might be unfamiliar with this office, it's a federal office within the US Department of Interior, where we strive to provide objective scientific data that data managers, policymakers, and you all, the public, can use to make informed decisions about our natural resources. So the USGS is Alaska Science Center is not a management organization, and we do not make any policy or regulatory decisions. We just provide information. So at the Alaska Science Center, I serve as the director of the Molecular Ecology Lab. So the Molecular Ecology Lab provides genetic information on the health and status of biological resources. So in our Molecular Ecology Laboratory, we conduct research projects that pertain to wildlife health and disease, population genetics, and also environmental DNA. And we've been conducting research on avian influenza in wild birds in Alaska in the Molecular Ecology Lab for approximately 20 years now. So when we began studying avian influenza viruses in the Molecular Ecology Lab, the ecology of these viruses closely remembered what's depicted on this really busy slide. So I'm not going to cover all this information, but I would like to step through just a few portions of the slide to describe where HPAI comes from. So not all influenza viruses affecting birds cause HPAI. There are many types of avian influenza viruses that naturally occur among waterfowl, shorebirds, and gulls, and these typically don't cause any disease. Birds usually appear perfectly healthy when they're infected with these types of avian influenza viruses. And we have repeatedly found these types of avian influenza viruses in wild birds sampled throughout Alaska for many years now. So these avian viruses that naturally occur in wild birds sometimes spread to poultry. Usually poultry that are infected by these viruses also appear perfectly healthy. It's only a small proportion of avian influenza viruses that make poultry sick. And it's this small proportion of viruses that cause severe disease and death among poultry, specifically chickens and turkeys, that are referred to as HPAI viruses. And so the tendency for these influenza viruses to cause serious disease and death among birds or to become highly pathogenic, that typically only occurs after viruses from wild birds have been introduced in the poultry. So these HPAI viruses that evolve in poultry are readily spread among chickens and turkeys. So these viruses may result in the death or destruction of very large numbers of domestic birds, and so therefore they're very costly to the poultry industry. So for example, since just 2022, more than 200 million domestic birds have been affected in the United States alone. So the spread of HPAI from poultry to wild birds, depicted here with the red arrow, has only become common since 2002. That is, historically, HPAI was exclusively a poultry disease. So for example, prior to 2002, HPAI had been detected exactly once among wild birds, specifically in terns in South Africa in 1961. Things have changed. So over the past 2 decades, HPAI has become an important wildlife health issue. Detections of HPAI among wild birds have become much more common, and this is particularly since 2021. So underpinning the emergence of HPAI as a globally important wildlife disease is a relatively recent evolution for these contemporary viruses, such that they're now readily maintained and dispersed by wild birds independent of poultry. And this has facilitated their rapid geographic spread. HPAI has recently affected wildlife in remote global regions with very few or no domestic birds, including locations within the Arctic and the Antarctic. Australia is the only continent on which wildlife have not recently been confirmed to be affected by HPAI. Also, HPAI is no longer strictly an avian disease. Many wild and domestic mammals have been affected by HPAI. Although relatively rare, HPAI has also affected humans. So the unprecedented spread of HPAI since approximately 2021 has resulted in significant impacts of disease to wildlife throughout the US and also globally. So for example, in the US, there have been extensive reporting of disease and mortality among diverse raptors and seabirds. Species adversely affected include bald eagle, black vulture, California condor, Caspian tern, rough-legged hawk, and peregrine falcon. Depicted here are some examples of excellent scientific papers that convey HPAI-induced mortality associated with these species of birds within the United States. Globally, there have been numerous reports of very large-scale mortality events associated with HPAI infection among wild birds. So, for example, during a single outbreak event in late 2021, leading into 2022 in the Hula Valley of Israel, it was estimated that many thousands of Eurasian cranes, or roughly 20 to 27% of the birds using that particular non-breeding site, died on account of HPAI. As part of a mortality event in eastern Canada during 2022, it was estimated that at least 40,000 seabirds died from HPAI, including large numbers of northern gannets, common murres, and common eiders. In South America, it's been estimated that more than 600,000 seabirds and more than 50,000 marine mammals died on account of HPAI following the introduction into the region in late 2022. So to further illustrate the recent spread of HPAI, I'm now going to briefly review the number of confirmed cases within various sectors in the United States since just 2022. So as of 30 May, there have been more than 19,000 confirmed detections of HPAI among wild birds in the United States, with cases in all 50 states. There have also been approximately 800 confirmed detections among wild, feral, and non-dairy domestic mammals. I want to point out here that these confirmed detections among wildlife in the United States represent only a small fraction of the true number of wild birds and mammals that have really been affected. And as per domestic food animals, more than 200 million domestic birds have been affected by HPAI in the United States since February 2022, including poultry in all 50 states. And HPAI has also been confirmed among more than 1,000 dairy cattle herds from 20 states. So that's herds, not animals. And also, as the number of cases in HPAI have mounted in wild and domestic animals throughout the US, so too have the number of cases reported in humans. So the CDC has confirmed 71 human cases of HPAI since 2024, the majority of which have been associated with domestic animal contact. And the CDC does report 2 deaths associated with those cases. So there have been many new hosts and routes of transmission recently identified for HPAI viruses, presumably on account of both increasing exposures as well as ongoing viral adaptation. So the ecology of HPAI has correspondingly become extremely complex as depicted by this slide. Thus far, the HPAI situation in Alaska has been somewhat less complicated and in many ways less severe as compared to other regions within the United States and elsewhere in the world. So this is at least partially a function of limited poultry production and dairy cattle production within our state. So this slide helps to summarize the current ecology of HPAI viruses and wild birds and spillover hosts here in Alaska. So HPAI viruses are now readily maintained and dispersed by wild birds. HPAI viruses have sometimes spread from wild birds back to domestic poultry here in Alaska. And HPAI viruses have also sporadically spilled over into wild mammals. HPAI viruses have caused some level of mortality among virtually all animal species in which they've been detected. So far, mortality among wild birds and mammals affected by HPAI has been reported to be sporadic in a fairly low magnitude here within Alaska. So, for example, HPAI has been confirmed among a total of 576 wild birds as of yesterday. More than half of these detections have been among ducks that appeared perfectly healthy when they were sampled or harvested by hunters. Of the birds in Alaska that have been found to be sick or that have died on account of HPAI, the more common species include bald eagle, common raven, common murre, glaucous skull, and glaucous skull. And other birds that have been repeatedly reported to be adversely affected in Alaska include black-billed magpie, black brant, Canada goose, lesser scaup, northern pintail, parasitic jaeger, savings gull, and short-tailed shearwater. So HPAI has also been confirmed among a relatively small number of wild mammals in Alaska, and this includes 7 red fox, 1 Arctic fox, 3 polar bears, a black bear, a brown bear, an ermine, and a mink. All these wild mammals that were confirmed to be positive for HPAI were reported to be sick or to have died. So now let's focus on what we know about HPAI affecting wildlife locally here within the Nome area. So as reported to me as of last week by the state wildlife veterinarian, Dr. Kimberly Beckman, the Alaska Department of Fish and Game has submitted 126 samples from Game Management Unit 22, and these were collected during June of 2022 through May 2026 and subjected to HPAI testing. So the animals tested include Arctic fox, a coyote, an unidentified eider, an ermine, a mink, a moose, a muskox, a polar bear, red fox, and a walrus. So HPAI infection has been confirmed in samples from 4 animals submitted by ADF&G. This includes 2 red fox, a mink, and the unidentified eider. And HPAI positive samples from 3 of these animals were collected relatively recently. That is during February and April of 2026. So in addition to the 4 positive HPAI cases confirmed through the Alaska Department of Fish and Game submissions, there have been 3 additional confirmed detections of HPAI in birds inhabiting the Nome area. Specifically, 3 glaucous gulls sampled by Gay Sheffield and others in 2022 are reported by the USDA to have tested positive for HPAI. These birds were also reported to have been sick or to have died. Some of you may recall that Dave Sinnott of the USDA was here last year and tested subsistence harvested birds around Nome. This occurred last spring. So earlier this week, I touched base with Dave, and he reported to me that he collected samples from 249 birds during 8 to 27 May 2025. And these samples originated from diverse waterfowl, including American widgeon, black brant, black scoters, cackling geese. The laundry list is here. You can read the species and the samples. Sample sizes, that's what the N numbers indicate. But in short, there was no evidence for HPAI among any of the hunter harvested samples tested. So in addition to information on the occurrence of HPAI among Nome area wildlife, we also have genomic information that's been useful for characterizing the viruses causing disease. So by genomically sequencing HPAI viruses from Alaska wildlife, we've been able to infer where these viruses may have come from and to estimate when they were likely introduced into Alaska. So this figure on the left indicates how frequently different types of HPAI viruses were detected based on the height of these colorful stacked bars. And the colors of the bars correspond to different regions in Alaska as depicted to the map to the right. So results specific to Nome are indicated in light blue. So the timeline below the colored bars indicates when different types of HPAI viruses were estimated to have been introduced or to have emerged here within Alaska. So as you can see, 2 different types of HPAI viruses were detected in Nome area wildlife during 2022. These types of viruses are referred to as A3 and B3.2. Both of these types of HPAI viruses were widespread in Alaska during 2022. Type A3 viruses were first introduced from East Asia into Alaska, and we estimate that to be in late 2021. And type B3.2 viruses were first introduced into Alaska from Canada or the lower 48 United States in early 2022. We also have some information on previous exposure of apparently healthy animals inhabiting Alaska to HPAI viruses. That is, we've been working with various partners to look for antibodies that react to these HPAI viruses in live wild mammals sampled throughout the state. So as part of these efforts, we tested 21 red fox serum samples collected during 2023 from the Nome area, and specifically this was Game Management Unit 22C. We found antibodies reactive to HPAI viruses in most of the red fox samples that we tested. So this means that red foxes within this region have likely been exposed to HPAI viruses and survived infection. So to summarize, recent research efforts on HPAI within wildlife of the Nome area, I'd like to follow 5 takeaway messages. So first, various local, state, federal, and academic partners in Alaska and elsewhere have been sampling for and conducting research on HPAI viruses in wildlife within the Nome area, also other areas throughout the state. Second, numerous HPAI viruses have been detected from sick and dead wild birds and mammals inhabiting the Nome area since approximately 2022, and this includes several recent detections. Third, multiple types of HPAI viruses have affected Nome area wildlife, and these viruses were introduced from both East Asia as well as elsewhere here within North America. Fourth, antibodies reactive to HPAI viruses have been identified among live mammals that suggest that all animals do not die of infection. And finally, HPAI may now be an established wildlife disease within this area, and it's unlikely to go away anytime soon. So I'm now going to pass things over to Lisa so that she can introduce herself and her work and provide more information about this ongoing sampling effort as she's leaving.
And we'll hold the questions till the end, huh?
That's good. Is that good? All right, thank you very much. It's a privilege to be here in Alaska. My name is Lisa and I am from Memphis, Tennessee. And I work at St. Jude Children's Research Hospital. It's my first time in Alaska and just came straight to Nome because I've worked with Andy in the past and through a lot of recent communications, we've been trying to figure out how to collaborate a little better. So I will start by telling you why in the world is a children's hospital working on avian flu? It's because of this gentleman in this picture. This is Dr. Rob Webster. He's been working on avian flu for decades and decades. And in the late 60s and early 70s, he came to St. Jude as a faculty in infectious disease. And he brought with him an idea that maybe the birds have the flu. And for a long time, no one really believed him. They're like, oh, probably birds have some flu, but does it really matter? We don't know. So he talked about this for a long time. And then it wasn't until the 1990s when high-path flu really kind of blew up in Asia that everybody kind of did a sit-up and say, oh, I guess bird flu is a real problem in the birds. And Dr. Webster was correct. And he what that did was when that happens in research, what they do is they throw a whole bunch of money at you. And so that was really fortunate for St. Jude. And so Dr. Webster was able to. He started a World Health Organization collaborating center for influenza research at St. Jude. He was able to get a lot of large grants, including now we are funded by the National Institute of Allergy and Infectious Diseases, which we have been for about 25 years now. So he's kind of the impetus behind it all. And the reason it's important for St. Jude to do this is because we are a hospital that treats children with catastrophic diseases, and that's a specific kind of hospital. We're not a regular children's hospital where kids with broken legs or something come in to get treated and they go away. These are children with life-threatening cancers and other life-threatening illnesses, and this is anytime from babies all the way up to 18 years old. And so that is the focus of the hospital. And when you have children with catastrophic diseases like that, most of the time they are treated with immunosuppressive drugs to treat cancers. And when you have that situation, the infectious diseases become really an issue. Like a lot of times people can get the flu or other respiratory illnesses, you get sick for a while and then you get over it and it's okay. Not these kids. If these kids were immunosuppressed and were to get even a common respiratory illness, this could be a really severe outcome. So they have put a lot of money and effort at St. Jude into the study of infectious disease, including acute respiratory illnesses like influenza. And the reason bird flu is tied into that is because of Dr. Webster's longtime work. He's not the only one, but he was one of the pioneers linking flu and birds as the reservoir for flu. So give you the mission of our hospital, started by Danny Thomas, who's an old entertainer that I don't know who I don't know, Gay, if that's in your repertoire.
I'm that old.
Okay. But he was an entertainer back in the 60s and he He was the one that started the hospital, and the mission is no child shall die in the dawn of life. It is that simple, and we don't ever waver from that mission. And like I said before, we've been a WHO Collaborating Center for the Study of Ecology of Influenza Viruses in Animals. So we're very specific to the zoonotic part of this disease. Flu that is in animals that can then transmit to other animals and then can also transmit to people. It's also important to remember, I get asked a lot about why do we care about flu in birds? Who cares? Is that every pandemic in history, going back to 1918, does contain an avian genetic component. So what that means is the genetics of the virus that Andy talked about is a little bit complicated in terms of viruses. Most viruses have maybe just one piece of RNA or DNA. Flu has 8 pieces of RNA. And when you have different viruses get mixed up in birds, all that genetics gets mixed up. And that can actually mix with viruses from other animals like swine and then human viruses as well. So the birds are really, really important. And they are sort of like the starting point for how flu evolves through birds and through other species. And so we have had now decades of global influenza surveillance at St. Jude. We're part of a network called the CER down there stands for Center of Excellence for Influenza Research and Response. We're part of a big network of other institutions that's funded by the NIH to do flu research. And so what does that mean for me? So my job is to direct the avian influenza surveillance program within our lab at St. Jude. And again, I told you we're funded by the National Institutes of Health, and this is kind of what we do in terms of research. We do a lot of surveillance that is not intended to be the same as what the USDA does or what even USGS does. Yes, we're out there, we're surveilling viruses, but we like to take that information, then turn it into real research projects where we can understand the disease better. And so that's what my job is, and I'll get into bird watching, what that means in a minute. But the first aim of our study is to expand this network of surveillance. So surveillance, if you think about it, it's usually very regional, and it's done, especially wild bird surveillance. It's directed and it's regional, and sometimes it's not connected. You know, birds are flying down flyways, up and down flyways all the time. And we just have kind of spotty surveillance that then over time does give you a pretty good body of evidence. But at the time that things are happening, you don't get a lot of information at the time the birds are moving. You usually get that information a lot later. So our first job in aim 1 was to target species and then target the flyways and get that a little bit more enhanced. Our second aim is to study viral evolution and adaptation. So we're going to take those viruses we get from doing our surveillance, and then we do genetic sequencing, and we try to figure out what the difference is between the high path and the low path viruses. Andy talked a lot about high path viruses. They're extremely important in a small proportion, but there's also a ton of low path viruses that these birds circulate too. And when those viruses get mixed up with high path viruses, usually from different regions of the globe, that's when you might get a new strain of virus, which is what you talked about, which in the situation we're in today, that's exactly what had happened in late 2020. And so we're looking for what's called reassortment, which is that third virus down there. That's when I told you the genetics gets all mixed up and then you get a different virus. Sometimes the virus you get is not as pathogenic as the first one. Sometimes it's the same and sometimes it's worse. And so that's what we're looking for. And then the third aim is we're going to take that information, and we really want to try to understand the bird better. We don't understand the immunology of birds. We don't understand, they're different than mammals. And so there's a lot of tools and technology that's available. I'm not going to go into it today, where we can take samples from wild birds and we can really study not only what they've been exposed to, but how the genetics of their immune system is working, and how that differs between species and how they can manage the disease. So what are the goals of Birdwatch? Birdwatch is a mobile scientific testing facility. And so just to keep it real, what that means is I drive around in an RV that is mostly held together by duct tape and mud at this point, because I've been driving a lot of miles in the lower 48 lately. And we go there, and I will go to a site where they are capturing birds, or we go to a hunter harvested situation, lots of situations where I can get my hands on some birds, and I will swab the birds. And then I will do a molecular test on site in the camper.
Mhm.
And so that's a little bit of a different twist on how you do surveillance. Usually our surveillance programs in this country have been completely retrospective. You go there, get the samples go back to a lab, A, B, and C happens, everybody announces, and then a few months later, you will get a result. That's great. There's nothing bad about that at all. However, what it doesn't give you is information that's happening in your community at that time. You get it later. But that's okay. But if something happens, because the thing about influenza is it changes so rapidly that it is really difficult to keep up with. We're always behind the 8 ball with viral evolution and flu. And so this tool is designed to just give you a test that will give you an answer rapidly. And so we can talk about the implications of that later. It's not appropriate everywhere, but one place it could be appropriate is in a place like remote Alaska, where you don't have a lot of resources and you don't have a lot of support out here, at least from your larger governmental organizations. You certainly have plenty, which is what Andy talked about. But if you have a disease now that is so prevalent in wild birds and things that are changing faster, you might need to think about, would it be cool to have information faster, one thing, and to be able to have a organization that could come to sort of fill in those research gaps that you might have. Again, not to do the same thing as what everybody else is doing, but to take it and then amp it up. Like, let's figure out some questions. Let's ask some questions and then use resources so that we can figure out how the disease is working in these birds in this area. So that's kind of the point of why I came to Nome, was to just test this out as a pilot to see how it might work. This is keeping it real. So this is me and this is what I look like most of the time. I live in this camper about 6 months out of the year. It's not very big, it's about 25 feet long. It's called a toy hauler. So a toy hauler, if you're not familiar with RVs, I live in the front of it and then I work in the back. It's built out like a garage. And so you're supposed to be able to drive your ATV in it. Like normal people would just drive their ATV and then it's a toy and then that's what it's supposed to be, it's for fun. So my toys are expensive instrumentation for laboratories and a lot of plastics and stuff. So we built that out of the back. The bottom picture there is what it looks like on the inside, but I really cleaned it up for the picture. So it doesn't normally look that clean. And then that's me in my all my disguises doing the actual test in the lab. That's like some professional photographer took that picture, honestly, that it really isn't that dark in there all the time. And that's an example of what I've done. This particular example is when I work in remote Alberta, Northwest Alberta. We have a site up there we've had for a long time. And I started bringing the camper up there and doing the testing on site. And this is just an example. Mostly they catch mallards up there for preseason banding. And so that's just an example of last year. We tested 610 birds. I got 105 positives right there on site. And what that allows us to do is do this at scale. So we can do that. I can pretty much throw out the negatives so we don't have to bring all that back to the lab. And then we just take the positives and work them up. So that's what it looks like normally. So Nome, I learned I couldn't drive here. Early on, I could not drive the camper here. So I packed it all up and put it on a plane instead, which was kind of fun. And so this is where we are now in Nome is me. And that's the picture of it all packed up in the suitcases. And that was me the other day with the sign that Andy helped me make. That's on the rental Jeep. So, and that's at East End Park. So the goal here, the mission, is to see if we can sample some hunter harvested birds like Dave Sinnott did last year and see what happens. Because sometimes what happens is people get a little complacent. Like, so, Dave's results were all negative. And so people might say, well, so what then? Why are we still doing this? That's not how flu works. So just because you didn't find it one year doesn't mean it's still around. Andy just showed you a whole bunch of data saying that it is still around. One sampling effort is great, but it doesn't tell the whole story. So it's good to be, the best thing about doing surveillance is to just kind of be strategic about what you do. You don't have to sample every single bird and be consistent about what you do. And I think one of the issues of Nome, what I'm learning, is that you may not have the consistency you want in terms of the sampling and the structure around you to get those things tested in a timely way. So that's kind of another reason why we're here to try that out. So these are just all the influencers that I work with. Birdwatch has sort of taken off. And it's a little crazy. And now we're out here in Alaska, so it's so exciting. But these are the partners that we currently work with. I can't wait the next time I make this slide. I can add some more partners to the Alaska one down there at the end. So I am gonna stop there and turn it back over to Andy because he's gonna do the final for you. So you can
Bring it home.
to do. Yeah, bring it home, Andy.
So to wrap up here tonight, I just want to make sure that we mention some resources in case anyone finds these helpful. Maybe some sources online where you can find additional information as well as contacts if you observe sick or dead wildlife. So if you want more information about HPAI detections in wild or domestic animals in the United States, you can find comprehensive lists of all confirmed USDA laboratory detections online at the address listed here. Per Lisa's comments, just be aware that often the results posted online are weeks or sometimes months after samples were actually collected. And if you want to become more familiar with information about human health and safety on account of HPAI, I encourage you to check out the CDC website. They have various websites, but this web address is a particularly good one. It has links to some other websites as well. And I appreciate, might not be able to write down these addresses right fast, but Gay's recording this. And so you can find these addresses online on that recording.
I'll try to call it up, drop it in the chat.
Awesome. If you are a hunter, as most folks are here, or help to process wildlife that's been harvested, so USDA and U.S. Fish and Wildlife Service have guidance online with steps that you can take to reduce the risk of HPAI to human and also domestic animal health. There's numerous suggestions offered by these agencies, but some common and consistent ones are disinfecting knives, equipments, and work surfaces that were in contact with wildlife during butchering, as well as cooking meat to an internal temperature of 165 degrees Fahrenheit to kill the virus. And again, USDA and Fish and Wildlife Service guidance for hunters can be found online at these very long URLs indicated here. And then finally, if you observe sick or dead wild birds that you think may be affected by HPAI, I encourage you to call the U.S. Fish and Wildlife Alaska Sick and Dead Bird Hotline at the number listed here. This goes to an Anchorage office. Observations can also be reported on the LEO network. The LEO network is a great way to record what you're seeing. It doesn't initiate a response necessarily, so just be aware of that. If you want to report sick or dead wild mammals, I encourage you to please contact the Alaska Department of Fish and Game. And if you're unsure exactly whom to contact or have problems reporting observations here in the Bering Strait region, I encourage you to contact Gay Sheffield. So Gay has been helping with HPAI response since mortality began here in 2022, and she is an absolute champion for your region. So contact Gay if you have any questions or observations. Thank you to everybody for your time and attention this evening. And Lisa and I would be happy to answer any questions that you may have.
All right. Thank you so much. Here we go. Thank you so much, Andy and Lisa. You should come on up in case we have any questions from the audience. But before we do questions, this is a good time for everyone while they're thinking of their questions to show some love in the chat. Or I'm not an emoji person, but there's lots of people who are. And show some love in the chat for our two speakers who've come a long way to provide some surveillance for us in the region and tell us more, especially learning about the your slide with the new Bering Strait information. So we really appreciate you guys taking your time out when you could be monitoring, or they could be out in the sunshine. We're really having some good weather here. I know I've got questions, but everyone else can't beat timely and in person. Thanks very much. Thanks, Trees and Dirt. And any questions? I guess first off, we have a caller, and I always am soft on callers because you can't raise your hand and you can't unmask yourself to be seen, really, at least when I was doing these things. So is there any question from the caller? question, comment? If not, that's okay too. And I think it's *69 if you're like me on how to get somewhere.
Yes. Hey, Gay, can you hear me?
Yeah,
Yeah,
We can
this
hear-
is Travis Booms from Fairbanks, Alaska. Just great job. I enjoyed the talk.
oh, wonderful. Thank you, Travis. Wish you were here.
Me too. I'll be there in a couple of weeks.
All right, let us know. And is there any update from the Fairbanks region on avian influenza?
I don't have any of that information. What I can say on a somewhat related note is that we did a raptor survey in May with some of the people that are there at the Peregrine Fund, and the raptors are doing very well this year. I know that's
All right, well,
only tangentially
hopefully,
related. Now, that's what I have to report.
awesome. Well, hopefully we'll get them to do a straight science later on in the fall and they can tell the region all about what they've learned. So we really appreciate that. And you, Peregrine
You bet.
people. All right. All right. I know, any other questions?
I have a question.
Yeah.
Do any of your collaborators do testing themselves, or are they always bringing you birds and you have like a whole lab set up? Like, depending on like how complicated your setup is there ever a case where you can have other collaborators also doing the testing like remotely?
Not yet. Working towards that. That would be amazing. But right now we do, I'm at least as far as I know, there could be someone else out there I don't know, but as far as I know, in terms of somebody that's out there and is so public facing as I am, then I don't know of anybody else that's doing any mobile testing. But that's not, I'm not tuned into everything. So, but yeah, as far as I know, it's just me. I'm sort of we call it the traveling sign circus. This is what it is right now.
And I don't know if you mentioned, but you're here until the 8th.
Until the 8th, correct.
And she's at East End Park and will be swabbing anybody's harvested eggs, birds.
Mhm.
And I would say you're open to harvested eggs and birds.
Harvested eggs and birds. Yes,
Okay.
that's what I have capacity to do
All right,
right now.
wonderful. And I also should mention that, you'd mentioned we're low in services out here, and that's why we're so glad both Andy Ramey from Anchorage has taken the time to make, and the money to make the trip out here, and yourself. And I want to give a shout out to the collaborative nature of response now in the Bering Strait region, because it has really, there's been a lot of pressure on this region from climate, from you name it, ecosystem changes, industrial changes, geopolitical changes in our region, and or in the area, not in the region, but there's a lot of things happening around here. And I want to give a shout out to the Norton Sound Health Corporation, Office of Environmental Health, COERIC, Natural Resource Division, Northwest Campus, which is lending you the lab space in this room, my office, the Alaska Department of Fish and Game here in Nome, Region 5, and Nome Eskimo Community. And every one of those, and if I've left anyone out, I'm really sorry. Those here in Nome are kind of the groups that are all collaborating as a team and bringing you in and sort of helping you get your feet under you here. And also a shout out goes to every single person who is utilizing our natural resources throughout the Bering Strait region. The animals here, because people already know this in the region, but the animals here are, we have comprehensive use of the marine and land ecosystem for food. And because of that, when we hear of trouble, and it looks like this bird flu can cause trouble to people as well as, we're seeing it in the animals. It's a food security issue. It's a human health concern. It's a conservation concern because we want big populations of animals, healthy animals to utilize and to have. We want the animals living a healthy, happy life. So I just wanted to add that and thank you again for coming here where we have so infrequent check-ins. We really, really appreciate that, and you working with the team of people here in the Noam area, which is also responding to all our 16 coastal communities and 20 tribes, and every single person who's a general public or a resident or visitor here. So we're really appreciative of that, because it's a big topic if anything goes wrong in our region. Any way you could comment on marine mammals? That's where I'm interested. You did mention polar bear. Is there anything we should be looking for? I actually got a question today. Is there any symptoms that we may, or behaviors that we may see in the birds or the marine mammals that might give us a clue to that there's something we should or shouldn't do, or something we should video or take a photo of or report for the birds? Or for your, like you've got depression on the slide there, paddling, paralysis, neurological. Is there anything you can speak to on that? What to look for? Because we're out here handling lots of animals throughout the region.
Sure, I'll take a stab and Lisa, you can fill in the blanks. So yeah, excellent question, Gay. And per this really smart question, there have been many marine birds that have been affected. And so there's, when birds are interacting with mammals, which, seabirds and marine mammals obviously are sharing habitat, certainly transmission could occur. There's now been 3 confirmed cases of polar bears in Alaska. So we do know that marine mammals in Alaska are being affected. There has not yet been a confirmed case in Alaska of any pinnipeds affected. So I want to clarify that
Yeah, we're glad of
been
that.
confirmed to date. But like what we saw in South America, there was many marine mammals infected down in South America. In fact, the virus in South America evolved. It was going marine mammal to marine mammal. Again, that has not occurred here in Alaska, but it has occurred and caused really large mortality events throughout several countries in South America. So things that were clinical signs that occurred in those mammals included things like depression, so just animals that weren't very responsive, maybe had some labored breathing, I think ocular discharge, so had some fluid kind of coming from the eyes or from the nose. I think that would be another symptom. Neurologic signs, if there's, little tremors or seizing, that comes to mind as something else to be looked for. But, I think folks here are really plugged into animals and animal behaviors. So I would just say, general rule of thumb, if, local residents in the Bering Strait region see a marine mammal that they think is acting abnormally, I encourage them to please go ahead and report that, call that in. And then, if possible, to kind of keep an eye on the situation, provide updates to, again, these contacts. And then hopefully there can be a response. There's kind of formal channels where people have their jurisdictions to go through. But again, if you see something that seems abnormal, I think it's probably worth reporting.
If you see something, say something. That's what we do. It's the easiest thing to spot is the neurological signs, and that is true in birds and in the marine mammals as well, from what we can tell. Just acting not right, and the lethargy. So I am a casual wildlife observer, right? I'm not a professional by any means. And I can easily tell the difference if an animal is lethargy or the neurological signs. So it's not difficult to pick up when they're sick. So yeah, just emphasize that.
All right,
There's questions back here.
another question.
Yeah.
Sorry, go ahead.
It looks like you have big marine mammals affected, like walrus and polar bears. Have you had any cases where it's been tested in whales?
So again, globally, there's 3 cases in Alaska, and there was now recently a 4th case just maybe a couple weeks ago. There was a polar bear case in Svalbard, Norway. Biologists in Svalbard. I've also now confirmed 2 cases in Atlantic walrus for high path avian influenza, these newer ones that typically cause disease in birds. That has, to my knowledge, not been detected in a whale. It has been detected in the dolphin in Florida. Other types of influenza, the ones that typically do not cause disease, have been detected in whales many years ago. So I think it's possible that a whale could be affected, but that has not been confirmed recently, to my knowledge. Great question.
Probably not much testing, I would guess, too.
Probably not
Because
much,
it
yes.
should be learned.
Yeah, there's opportunity for sampling there during hunting season.
Yes, during hunting, but sort of the rest of the United States, yeah. Good question.
Great, great comment too.
There's a question in the chat. Given that we are in a major pathway of bird migrations, and that would be coming at us from the Russian side and also the US side, you're gonna have to make your maps a little bit more western looking. Why has the testing for bird flu been so lagging?
That's like,
Which one of you wants to
I don't
take
know.
that? And I'll preface this by saying in 2009, we were really well trained by the USDA. Do you guys all remember 1-800-CALL bird flu? Yeah, see, Molly right there, 1-800. Now I know that's probably ancient history for you guys, but they did a really robust job. We had people that came here and went out to St. Lawrence Island and whatnot. So they were just looking for it at that time. So it is a question, and I appreciate our person who wrote that in. We are a major pathway. Why has the testing for bird flu been so lagging?
I'll take a stab, and it's true that Alaska is extremely important for migratory birds. I think we can all probably appreciate that. I think it's true, per your comments, Gabe, that back in 2009, there was much more testing. There was much more political appetite to fund avian influenza testing. I was actually testing birds back then. I saw it myself. We had a lot more capacity. We're at the time, I think, that people can appreciate where it's doing more with less, or maybe sometimes less with less. There is still a fair amount of testing going statewide. So I provided the number of detections today, talked about Nome area. There is a good bit of additional sampling. USDA does have a program. Dave Sinnott, whose name I mentioned for USDA, is sampling a good number of dabbling ducks primarily, perhaps some other waterfowl. The sampling efforts typically in autumn. And to my best understanding, that sampling activities takes place in autumn because that's when viruses tend to be most common. So there's lots of young, I think we can all appreciate that. They hatch, and as those young birds become more mobile, they don't have very developed immune systems. They tend to be very susceptible to disease. And so the best bang for our few dollars is in autumn. That's when we're most likely to detect these high path influenza viruses. I also lead a research program where we conduct pretty intensive surveillance around Eisenbeck Lagoon, the town of Cold Bay in southwestern Alaska. This, to my knowledge, is the most intensive surveillance activity that occurs in the state of Alaska. This will be our 16th year of conducting that sampling. We test approximately 1,000 birds there at Eisenbeck Lagoon in autumn. And so I'll be forthcoming. I've wanted to focus our resources at Eisenbeck because it's in autumn where we tend to get our biggest bang for our buck. And because we have this funnel of birds coming down from throughout Western Alaska, including Northwest Alaska, through Eisenbeck Lagoon. So although we're generating data from Eisenbeck Lagoon, we're also actually generating data from birds that are coming from areas such as here. And so I think, although the sampling occurs at Eisenbeck, why I didn't highlight it here, we're actually learning quite a bit about flu throughout a large swath of the state. And in past Straight Science, I'll put a plug for, there's 4 years of Straight Sciences that you can find on YouTube. And then I think all 4 of those, I include updates on what we found at Eisenbeck through the years. But we also have lots of publications on what we found at Eisenbeck Lagoon. I'm happy to send anyone that wants to contact me, Ramey at usgs.gov. I will send you publications we have, including one that's about to come out where we summarize 14 years of data. So we have a lot of information from that spot. This is all to say our surveillance isn't perfect. It doesn't cover the entire state. It's a far cry from what I wish it would be, but it's also not absolutely terrible. I mean, we do have a good amount of information from the state, and that's just birds too. We also have some information from mammals. But maybe for the sake of time, I'll leave it at that.
All right. Well, thank you very much. I'm going to try to grab your email here and drop it in the.
Do you have another question?
I had one last.
Yeah,
thing.
go ahead.
My question was, you said that you did the genetic sequences of diseases, and one of the things that you found was the presence of pieces of avian flu in all of them. What is it about the bird flu that sticks onto the diseases that spread?
Oh, that's a great question and really complicated, and I probably don't have a great answer for you, but I did speak a little bit about the concept of reassortment, which means lots of times there's so many different kinds of flu viruses circulating in birds. Oftentimes two different viruses will infect the same bird. And when that happens, you can get the genetics of those viruses mixed up, and then a different virus comes out. And oftentimes the avian genetic part of that will stick with the bird because it's evolved with the bird. So it likes the bird. And so that's why it doesn't get kicked out, or it's just like diseases that humans evolve with, right? There's lots of things that we cope with as humans that we've evolved with that
Birds aren't stuck in one place, they're always interacting.
they're always interacting. So
Okay.
that's kind of why, yeah. And that's not a great answer, and I wish we had a better scientific answer for you, but that's what we think is going on. Great question, thank you. Did you have one, Molly?
I had a potential two-part question and
Sure.
then an observation.
Uh-huh.
Kind of going off of what you were saying, was like, are you guys like doing just observation, or are you guys also trying to come up with like potential vaccine for at least like effective to like a current strand or something?
St. Jude does do that work. We do a lot of research back home in the lab on not only these viruses, but also other mammalian viruses, including human viruses. And we do suggest that, and we do a lot of vaccine consulting, I guess. My boss happens to be one of the people that goes to Geneva every year to decide on which strains of the virus will go into the next year's human flu vaccine. So we do study that quite a bit. What was the second? Did you have a second question?
If yes to that, the first
Yes,
one.
the answer to that is yeah.
So second part is, would you guys find a way to be able to put the vaccine safely into the waterways so that way wildlife would be able to get at least some of the vaccine?
That is actually, we get that question a lot. Like, why can't we just vaccinate the birds, right, and put it in the
Right.
water? I don't know the answer to that. I feel like technically, yeah, that's available, but logistically, and I don't know how that would actually work. Amy, do you know how to answer that question? Because I have buzzed this over my head. Also, I think
Take a stab.
Who's better at this than me?
So a couple things. This is an excellent question.
Yeah.
So one, in addition to the human vaccine, there are actually a few efforts that are pretty remarkable to develop wildlife vaccines. So, for example, California condors, if you ever heard of it, it's a bird that in the West Coast, in the lower 48, when I was growing up, there was down to, I think, 27 California condors. It's really a pretty remarkable conservation story. And now there's somewhere upwards of 500. But there was a die-off event where a fairly large number of California condors died. So they tried a poultry vaccine in California condors and actually vultures first. And it looked like it worked pretty good. And so now they're starting to put vaccines in captured California condors and hopefully provide that population with some protection. There's also another effort where they're vaccinating penguins in Antarctica, because that was a lot of concern about penguins in Antarctica. And so that's maybe a few steps behind, but it looks like maybe there's hope and that could be an efficacious vaccine. So I think in certain narrow circumstances, there may be opportunities to vaccinate wildlife. And I think that's an interesting idea to think about putting in water. On the other side, this is just a counterpoint, is that vaccines can be complicated because there's kind of this cat and mouse game with pathogens and vaccines. So if there's a lot of vaccine out there, then the pathogen evolves to evade the vaccine. And so if we vaccinate really widely, then we're also causing the pathogen to change really quickly. And so there's kind of a danger with putting out too much vaccine too. So I think in certain circumstances, as of right now, there might be some opportunities to vaccinate wildlife, but it's probably gonna be those populations where there's like not a lot of animals and like we really wanna be sure to be strategic about where we're putting vaccines and also the logistics, right? Because it's really complicated to capture animals and give them a vaccine, and sometimes it takes 2 doses, so you got to capture an animal twice, and like this might get really complicated. But like if we try to apply them too broadly, we might actually be making the situation worse by trying to make it better. So I think it's pretty complicated, but I think you're onto something. I think that's a a great question, and unfortunately, it's kind of a complicated answer.
Yeah, it has to do with the nature of influenza. Like if it was a different kind of virus that didn't evolve so quickly, it might be a different story. It might be a lot easier to do. But because flu changes so fast and it is very easy for flu, it's so good at adapting to any kind of immune pressure, right? Anything, it can change. That's why we can't get rid of it, right? That's why it sticks around and is such a successful pathogen. This is because it's so clever that way.
Question. You mentioned earlier that autumn is like generally the best bang for your buck time to test. But Lisa, you mentioned you spend roughly 6 months on the road. How are you just designing your? Is it 6 months straight? Is it on and off? How are you designing
it's not
that?
6 months straight, but I'm still in the build the plane while you're flying it phase, and so I'm out there trying to figure things out and trying to figure out how to be more strategic. So what I generally do is I start in the breeding grounds where the juveniles are getting ready to go, because that's what he said. That's where you're going to get a lot of virus circulating and in a big susceptible population. So we know we can monitor that in the breeding grounds. So that would be a place like Alaska, and I work a lot in Canada as well. And then I try to start that in like late summer, and then as the fall progresses, I just chase the birds down the flyway.
Just
right?
zigzag down the flyway.
Right, just zigzag down the flyway, and then by the time it's November, December, I'm in the very south part of Louisiana, when the birds are starting to winter. And so that's what I've tried to do. It's logistically a bit difficult, and again, I have to be zigzagging and going to where the places are that are capturing invading birds, so we can get birds. So
you're focusing on places that are actively capturing and banding?
Yeah, just mostly because that's where I can get my hands on birds.
Yeah.
It's just, how else do you get your hands on a lot of wild birds? There's the banding operations that are doing the management for the harvesting. It's a very complicated thing as well. But they've all been pretty generous. They all understand flu, and and it's easy when you've got a bird in your hand to do a swab, right? It's just, it doesn't take a whole lot of time, and then we can also do, by the way, we haven't talked about this, but we can also do sampling environmentally. So one of the things that is extremely successful is going out into an environment where the birds just were. So say there was a flock of, I don't know what, there's ducks or geese or whatever roosting somewhere. They lift off and go somewhere else.
Safety sound, let's
Okay,
use
safety,
safety.
okay,
That's local.
You can walk out there and start picking up the poop. So I do a lot of poop picking in the off time. Right? So we go out and you pick up the poop, and you can isolate virus straight out of there. So if you catch it right and you get some fresh poop, it's the same as getting the swab in the cloaca. So
Sorry,
keep going.
Yes. How fresh a poop are we talking here? Are you watching your phone? You got a little bit angry.
No, I tend to be on shorelines, like near the water, so it stays a little moist. It is an RNA virus, so you don't really want it to be out in the sun too long. UV light will do a number on it. However, it is protected in a whole bunch of organic material like poop, so it's not a huge crisis. But if it has all been like dried up after a day, then that's probably not worth it. But if you can go out soon after the birds have been done feeding or whatever they're doing and they've left, I can pick up 200 samples in an hour. It's great. So yeah, it's a real efficient way to do it.
Yep.
My question to you back would be, are you with your peregrine work, are you guys gonna be collecting some poop or swabbing or?
We collect poop for the deer falcons. We're mostly using it for microbiome stuff. But it's liquid gold for us. So like when we can get it, there's definitely multiple uses.
Could you take a little on a swab and?
Yeah, I mean, we also test for AI. We collect blood as well,
Okay.
so we mostly do it that way. But we've been doing that kind of off and on for about 10 years. We've only had like 2 confirmed cases
Are you talking here in the region?
here in the region. Yeah.
That's still very
In this region, Bering Strait region, you've
Yeah.
had 2 confirmed avian influenza cases in your
In Geological.
falcons.
And I think, I'm pretty sure that was during our fall trapping effort, so they were migrating birds and like, they might have been low path because the falcons were fine, it seemed like. And those are published papers, you might fact check.
Oh, I'd have to look that up.
I
So I should-
didn't see it in the list. Is it in the list?
Based on serum samples or is this
It would be
based?
serum, yeah.
Okay, so that's antibodies you've reacted to.
Yeah.
reactive to,
Yeah.
so it would,
Right.
may not have been an active infection, it's
That's
just what they
true.
were
Prior
exposed to.
infection.
So the poop would be, the fresh poop would be an active infection,
Okay, cool,
so
yeah.
that's it.
Okay.
Yeah, okay.
And then I think Molly raised her hand, right?
Yeah, keep
Um,
going.
You guys are all right with all this?
Yeah.
Okay, keep coming.
So during the bird mating seasons, the mouth would be a pretty good spot because there's like a bunch of tern nesting, or Arctic tern and Aleutian tern. And if you go all the way to Hastings, so that's like towards Cape Nolan.
Keep going, okay.
Yeah, and it's like 10, 11 miles out of town. A lot of seagulls and Arctic terns nest there too
Okay,
throughout most of the summer and then the fall time.
Yeah.
And there's like frost poop on them.
everywhere, yeah.
So if I can follow up to that excellent comment in the prior question or series of questions, I can gather my thoughts here. I had a couple things that I wanted to mention. One, I did make the comment about bang for the buck, which I 100% stand behind, but like not always is that the only factor, right? Like if there's a question where we have to sample at other times a year, maybe that's a good use of resources. I mean, I've like done that in some of the work in the past when, I was curious, back in the 2009-ish days about how viruses might move up from South America, and that required sampling blue-winged teal entering the continent from South America on the Gulf Coast. And so it was maybe a little less efficient for getting viruses, but we got really good information there about how viruses move between North and South America that had to occur in spring. So, if you want information on, when people are harvesting subsistence harvested birds and, that people are curious about what's the risk in the subsistence harvest, well, that has to occur in spring, obviously. So that's why it's here. So it's not only about bang for the buck. And, well, we're speaking about bang for the buck. I was thinking about, terns have been a very susceptible species and sometimes die. But like in my prior answer to a question in the chat, it was really talking about what's called active surveillance, where we're going out, catching birds and trying to get viruses. There's also this incredibly efficient system called passive surveillance, and that's also conducted not only here in Alaska, it's throughout the lower 48. And that's where you all are best partners. When people are out on the landscape and observing things and report them, and then we find sick and dead birds, do those reports, that's passive surveillance. And you all are participating with that when you make reports. And that's an incredibly great bang for your buck technique. So when you all are out on the landscape and if you see something that doesn't seem right and it's sick or dead birds and you report that and you're participating in this surveillance, and that's an excellent use of resource and we really appreciate that. So that passive surveillance is really important and I appreciate everyone that participates in that system.
Yes.
Would leaving sample kits in regions help you in your surveillance program? So people can send it into you?
That is an
Plus
outstanding,
information?
that is an
Context?
outstanding
On the way.
uh,
It's on the way.
question, and that's a little bit tricky one because there's some complications with liability when it comes to sampling, but I think that's an excellent suggestion. I think that is registered, and I appreciate that, and I think we've had lots of discussions about what that might look like. Excellent suggestion.
It is things that we think about all the time, and trying to develop that into something that is consistent and sustainable is completely region specific. So for instance, so like in a place like this, it's not that it's impossible, it's just that we would have to work really hard to figure out how it's gonna work and so that it works well.
Yeah, you could just leave kits at the National Wildlife Refuge or with the fishing game and then have like a free sticker of postal to wherever it's supposed to go.
Right.
Outstanding comment.
Molly, you had your hand up.
No, that's
okay.
exactly basically what I was going to say. Like we had a lot easier time in hub towns across the state, so that way like you have access to fishing gear.
And
Comments.
My question was, I think part of the lack of monitoring, you may be missing quite a bit. We know in 2022, this region was kind of lots and lots of reports of many, many birds, Safety Sound, St. Lawrence Island, birds that couldn't fly and twitchy and walking strangely or not responsive. So we had many, many birds and we reported all those. Do you think part of that picture of how it's like eagles were the most affected, those were at Dutch Harbor or Unalaska and in the more urban areas or in those refuges where you already have people manned and it's paid for? And we understand everyone's stretched, like you said, do less with less. We feel that too. But so there may be underreporting greatly than what it may look like currently, the picture of how it's progressed or where we are. And then we know the North Slope, when you were saying the animals fall time bang for the buck, the animals are moving down. We know that the North Slope Borough has their own Department of Wildlife Management, and they do a fantastic job up there as best they can, covering a very large area. And I'm not really quite sure. I think Kotzebue has the U.S. Fish and Wildlife Service there with refuge or
Sellewick
National Wildlife Refuge.
Yes.
Okay. Do you have any information on how those regions are either doing this year or have done in the past regarding, because they do have some monitoring, and what we might, I think about, oh, well, maybe they're getting it and we're going to see it in the fall because we're a place where those birds are coming down through here. Is that something that you think, are they indicating that they have a bigger problem like we did in 2022 at all?
There's a lot there. Let me try
Yeah,
to
yeah, yeah,
answer and then you let me know where
yeah.
I leave holes here. So 2022, I think, was pretty extraordinary, and we were stretched extremely
Oh, yeah.
for
You guys
a
were
couple reasons. I mean,
hampered.
one, something really changed in late 2021. I mean, it was clear the train was coming, to the point that I worked with folks nationwide, and we put out a report. We arranged a webinar series. We wrote a review paper that was published, I think, within days of the first detection in North America. The title of the paper is "Highly Pathogenic Avian Influenza Represents a Threat to Wildlife in North America." I mean, it was coming, and it came. And so even with trying to spread the word, the resources weren't there and ready to be mounted. I think we did okay. There was too many holes to put fingers in.
Sure.
And so there was a lot of detections across Alaska. We missed a lot. We know we missed a lot. And like those 19,000 detections across North American wild birds represents a small fraction of the number affected. There has been way more birds affected across North America than 19,000. Way more birds. And so like, I want to be clear here that is the minimum number, and that's like a low minimum. And per your comment, there's huge detection biases. An eagle's a very big bird. A swan's a very big bird. So like these are probably overrepresented proportionally because they're going to be found. And like small birds are going to be a lot harder to find. They're going to be reported a lot less. So take all this information and realize like there's biases in there. We know, we admit it, it's there. Also, there's this concept they didn't bring in today. We talked about it, some of the other straight sciences, of population immunity. So in 2022, HPAI hadn't been here in North America, not since 2016 was the last detection in a mallard in Alaska. But at that time, it was really, really low levels of HPAI in Alaska. So most individual birds, wildlife across the state, had never seen HPAI before and was completely naive to that disease. And so this disease, when it came fierce and it spread widely. Now, lots of these animals, the ones that are still alive, have maybe seen this before, like the data you saw from foxes. Most foxes here now, presumably, have seen HPAI, at least as of that was 2023 data.
From eating the dead birds that have died, or guessing that's the best way for them to get it.
Presumably, so if they have antibodies that reacted to HPAI, they're probably either they might be refractory from disease. That is, if they get exposed, they might not get disease. Or if they get infected, they might feel a little crummy for a short period of time and then get over it pretty fast. And so I think there's probably higher population immunity now. And so I think what happened in 2022 is probably less likely to happen again anytime soon. I
That's
think
good news.
there's probably building population immunity across the state. I think, it's kind of good news, bad news. A lot of animals are being exposed, but like maybe we're going to see less disease or keep this low level of disease. That also might have to do with the fact that we got like this low simmer that, I'm just being honest with people. I don't see how this goes away anytime soon. So just being honest with you, I think we might be in like the new normal now where it's like animals are getting exposed. Lots of them are surviving. Some of them aren't. So
I could take that and make that link again to the reason I'm here. So what he just described is a really awesome effort and classical ways that surveillance is communicated through scientific papers and through, government, and then it might trickle down to the national media. It's not always the way people get their information the best, right? So one of the reasons of having a mobile lab on site is for the communication to you regionally, right? It's like, okay, here's what's going on now. And we can tie that back to the larger research body that information, as great as it is not going to trickle down a lot of the times to the local people. So it's like you view us kind of like a bridge, like St. Jude's can be kind of that bridge from this huge body of work that is going on, and it's all true and all great, and everything about that is good. But then we can bridge that and bring it down to what matters to you guys here today in Nome.
Yeah, I think you're gonna work on birds as soon as we're done.
As soon as we're done, we're gonna go work on the birds that were collected over the last couple of years that have been in your freezer.
Yeah, that's I'm glad.
Yeah.
Do you look at the birds' body when you have asymptomatic positive testing birds? Like, do you look into like how that bird is evolving to respond to the disease?
Yes, that was aim 3. Way to go. Yes, that is taking those pieces of tissue, that is doing cellular analysis of what's going on with the genetics of the response to that bird. Absolutely. It's still sort of a science that is in its infancy, but what I have been doing is collecting those tissues when I can, storing them so that when we can actually get the right molecular tools to start looking at that, which they're coming online all the time, then we'll be ready to go with the sample set. So
That sounds exciting.
yeah, it is. It is really exciting.
Okay, and then last but not least, the online crowd's pretty quiet, I have to say. Or maybe you just get it in there on the chat. But when Lisa and Andy first came to Nome just a couple days ago, I took them down to the mass grave memorial for the 1918 bird flu. And that was very sobering for them because they were seeing, and you were mentioning, St. Jude Hospital, this is kind of that's a sobering thing. And it is, if no one's ever gone down there, you should. Is that, we hear about that, and it affected communities throughout this region, and then Shishmaref took the amazing strength to stop the forward migration of that disease moving forward at their own, I'm sure, their own loss in life there, but they were able to stop. That's a historically amazing story, and that's why we don't have it. It didn't go further north up into the North Slope and Northwest Arctic, but that was a bird flu. Is that right? That was a version of the bird flu.
It was.
Is that just to
there was
that
together? Parts of it that had come from birds, right? It was a human flu, but as those viruses, like I said, reassort, some of that avian genetics sticks around.
Right.
So yes, so there were birds involved, but I'm not
in the beginning.
In the beginning.
Okay, so that's a good example of why it's good to learn more about this
Right.
situation or this disease. So yeah. Any other No questions? No questions.
It's amazing.
Outstanding.
Outstanding.
I think the other one was the North Hillborough and Northwest Arctic. Are they,
No.
they're not swimming in it, are they, or do we know?
I have not heard swimming reports. Yeah, I have not heard swimming reports such that I anticipate something imminent.
Right, all right.
So you don't puzzle piece your information findings with the other regions?
Oh, I think yes, there's great communication. I'd say there's good communication among regions, and so I think folks do communicate and coordinate when there's even some. Usually before there's even confirmed findings, because these confirmation findings can take weeks or months. Often when there's a die-off event, people start communicating that throughout the state. So I do think there's pretty good communication. For sure, there's room for improvement.
All right, on that note. Anybody else?
Let's go.
All right, last call. Well, we'll let everyone go home and have dinner, and for those online, know that the next Straight Science will be Thursday, June 11th. And we will have Joy Baker, who is the Port Project Director for the Deep Draft Port of Nome. And that will be, we will get an update for 2026 on their scheduling and some of what we may anticipate here in Nome. And that will be the evening, same 6:30 start time, and that'll be Thursday evening, June 11th. So we'll see you then. And everyone have a good night online and in the room. So thank you very much.